Hello everyone, my name is Nicolas Roberto Villamil, and I'm going to present you our project called Transcriptomic Insights of Immune Challenge Colonies of Hydractinia symbiolongicarpus. First of all, let me present you our investigation model. This is Hydractinia symbiolongicarpus, is an Cnidarian and hydrozoan from the Fidifera 4 group, and have been described as a model organism from the Cnidarians because of its life history traits. It's a sessile and colonial organism like the gorgons, the corals, and our medusa. Also have been used in other biology fields like neurobiology, development biology, and stem cell biology. Hydroctinia has its whole genome already sequenced, and first characterization of its cell atlas, and a putative immune transcriptome. So, to perform the immune challenge, we selected a gram-negative and a gram-positive bacteria. We cultured them in LB medium at 37 degrees overnight until we got uh, 10 to the 8 cells. Then we prepared these two conditions: the treatment when we mixed both cultures, and the treatment when we use artificial seawater pasteurized, and five colonies each of this genotype of Hydractinia, and we place both in petri dishes with artificial seawater pasteurized. Then we expose the colonies to this treatment for 1 hour. Then we wash them with artificial seawater pasteurized, and wait 3 hours until we extract the RNA with the in vitro gen trial protocol. Then we purify it and send your RNA to sequence with DBNSEC from MGI in Jancell in Bogota, and then proceed to the bioinformatic analysis. Using HiSat2, we aligned our reads to the HISIM version 2.1 of the reference genome, and we got a percentage of alignment from 97 to 99% for all the samples. And we capture from the gene universe a 98.89% of genes. Also, we generated a cone matrix using HDSEC. Then we load the data into R and filter the conch matrix and perform this quality control. As you can see here, we in this PCA we got two groups: the controls and the treatments. This it's also reflected in both these of this analysis, the correlation map and distance matrix, when you can see the two groups. The only exception is this T5 sample but as you can see here in the PCA, the T5 sample grouped closely to the treatments than to the controls. So we decided to keep it for further analysis. So using this tool, we performed the differential expression analysis, and we got 1,325 differential expressed genes, from which only 922 were codificant genes. As a criteria for defining these decks, we used an FDR of 0.05 and a log change greater than 1 and lower than -1, as you can see here in this heat map. Also, we want to see how the distribution of the cell markers were among our decks, and for both the logarithmic regression and the Wilcoxon test cell markers. And we found that the conolipin positive cells, the gastric gland and the mucosal gland cells, were the ones that were positive expressed, with in the case of the logarithmic regression, the progenitors, the nematocyst nematoblast, the neurons, and the reproductive cells being the ones underexpressed. In the case of the Wilcoxon test markers, the progenitors didn't have a significant difference, but both nematocyst neurons and reproductive cells were the ones that were underexpressed. The epitheliomuscular and chromatin positive cells in both cases had no significant difference in the cell type, but yes, in the cell subtype. We also decided to perform this WGCNA analysis, from which we got this network of here. The colors that you see here are the ones from this correlation and significance analysis that allows us to see that these four modules, green, turquoise, cyan, and yellow, were the ones that significantly correlated to our treatment, and that these three of here, Pink, brown, and blue models were the ones that were not correlated at all. This was validated by the T student test, where you can see the distribution for the four related modules and the three unrelated modules. And as an example, we got this black module from the non-significant. Also, we wanted to see how our decks distributed among these models. We got a total of 586 genes in the related models, 524 in turquoise, 41 in green, 18 in yellow, and only 3 in cyan. For the not related models, we got 331 in blue and 33 for both pink and brown. 9 genes were not found, as in this were only 387, and as you can see, near of the most sharp genes. For the for see the cell marker distribution among our models, we perform these heat maps. And as you can see, there is no model with with a specific cell type associated. The most cell markers found were in the turquoise model, the blue and the brown, from the ones that have already described, and from green, yellow, gray, and red from the non-significant. In turquoise, in the case of the logarithmic regression, we got conniving positive, gastric cell glands, and mucosal gland glands with laser markers from adult cell types. From for the blue, we got nematocystis and nematoblasts with also a parallel distribution of the ulcer marks. And for brown, we got progenitors. From green yellow, we got nematocystis nematoblasts. And for gray, mostly neurons and a broadly distribution of the others. And for red, only a broadly distribution. In the case of the Wilkinson text, all the models got progenitor markers and epithelial and epithelial muscular markers with, as you see, the other expression that is found here. Also, we perform an enrichment analysis using the cluster profile packages and gene set enrichment analysis. From this, we decided to select the categories that we thought that were more related to the immunity, like this membrane category, or right here the mitochondrial respiratory chain complex, or the protein transport, and to the next analysis. Also, we performed this for the models, but we got the problem that some of the models, like these ones, got a similar proportion of annotated and unannotated genes. So only 11 of the genes of the models got an enrichment term or more. This allows to give these 11 models a characterization, and like in turquoise we got respiratory chain complex and lysosome moderation with other interesting categories like protease, serum protease secretion. And yellow got immune system regulation and stress response, and green cell matrix sedation, cyan nitrogen metabolism that were the ones related to immunity. And in the related models, in blue we got immunity, in brown we got cell cycle correlation and nitrogen metabolism, and in pink we got GDP signaling and endosomal moderation. Taking the interesting categories from the enrichment analysis and the gene set enrichment analysis, we perform a domain annotation using interpret scan. To this domain annotation, we perform a text filter of 255 text terms that we thought were closely related to immunity. And from this, we got these 107 domains. Interesting domains that we thought are related to immunity. And as here you can see the number of interesting domain proteins among our decks, the distribution of the categories among the enrichment unannotated and enrichment here is here. The majority of them were shared among the categories, and for the individual categories, lower numbers for unannotated and enriched, and the majority of them in unenriched. Taking the domain annotation from the related model decks, we were able to found these 41 more interesting proteins that we classified in four categories: complement system, effector molecules, receptors, signal and signaling molecules. Also, we got these two of here that were uncharacterized because the domain structure it's a little bit ambiguous. And taking this, we were able to give to some of the uncharacterized proteins from here a function. For example, this one that we think it's a complementary relation as its property like. And this one from here that we talked by its domain structure that it's a P2 related protein, as P2 is a cell cycle regulator and an immune system regulator. Here you can see the expression levels, where you can see that the majority of them were overexpressed in our treatment, and this group over here were underexpressed. Interestingly, here we found proteins with classical immunity-related domains as the TIR domain and the JAK signaling pathway. The distribution of these proteins in the cell types were mostly in the gastric gland cell types with some progenitors, epithelial muscular, epithelial, mucosal gland, and nematocystis hematoblast cells. And in here we add conolipin positive cells. Most of them came from the turquoise mold with exceptions of green and yellow, but none from cyan. And as a concluding remarks, we got that we were able to trigger the immunity from Hydractinia symbiolongicarpus by a bacterial immune challenge. We identified the immune response molecules. In this immune challenge of Hydractinia symbiolongicarpus, we found also both classical and non-classical immune-related proteins over and subexpressed. The domain analysis gave us the capability of assigning an identity to some of the uncharacterized proteins, and we thought that the immune-related cell types that appears to be related in this case were the glandular cells for the gastric system and the mucosa, and the conolipin positive cells. Finally, thank you very much for your attention, and thank you to the IGUN and the Universidad Nacional de Colombia for its support.